When we study about reproductive isolation, we come to know that the different species of animals and plants which exist on this planet, they are reproductively isolated from each other, and that is one of the ways by which the individual identity of a species is in fact maintained. So we can categorize this reproductive isolation into two major types. One is premating reproductive isolation and the other one post-mating reproductive isolation. One of the mechanisms under these two is sexual isolation or ethological isolation. We can study this aspect in two sympatric species of Drosophila, which are very common in Indian subcontinent, and these are Drosophila bipectinata and Drosophila malerkotliana. Actually, there are four different species which come under Drosophila bipectinata species complex. These are Drosophila bipectinata, Drosophila malerkotliana, Drosophila pseudoananasi, and Drosophila parabipectinata. These four different species come under bipectinata species group, and they are very much close to each other. They are derived from a single common ancestor. So we can study this sexual isolation between two phylogenetically very close and sympatric species, that is Drosophila bipectinata and malerkotliana. I'm going to explain about an experiment which is very good kind of experiment. We can plan this experiment in the laboratory conditions. We can show these experiments to our UG and PG students, and this will give the best idea uh, to know that how two different species are reproductively isolated from each other. See, as I told, Drosophila bipectinata and malerkotliana are close to each other. They are although two separate species, but under laboratory conditions, matings may occur between these two species. Although in nature, members of these two species never mate with each other, we have not been able to observe their hybrids because the hybrids formed from the mating of these two can be morphologically identified. So we have not been able to observe any hybrids so far from the natural conditions, but under the laboratory conditions, mating may occur between them and we may get their hybrids. So let us go through these experiments. Before going the real experiment we execute in the laboratory conditions, let us get acquainted with the morphological features of these two species. See, Drosophila bipectinata and malerkotliana females are very similar in their morphology. As a whole, we can say that these two species are morphologically very similar, but their females are very much same. We cannot identify any female of these two species uh, very confirmatory that this one is a female of this particular species. It is not possible at all, but their males can be distinguished. So this you can see in this diagram. This one is male of Drosophila bipectinata. This is male of malerkotliana. What we can see in case of malerkotliana, the abdominal tip of the male is black in color, whereas abdominal tip in case of bipectinata is brownish, and that is not black at all. So this is one morphological feature on the basis of which we can identify the males of these two species. As I told, their females are very much similar to each other. So it is not possible to distinguish females of these two species. Now, another identifying feature of the males of these two species is to observe their sex comb. Suppose in this diagram you see this one is sex comb of bipectinata. We know that sex comb structure is found in the tarsus portion of the prothoracic leg. So this is tarsus portion, and this tarsus itself gets subdivided into 5 segments. So in the first subdivision, you can see two distinct sex comb. This is one. This one is the second sex comb. You can exactly count the number of hairs also. Here 5 to 6 hairs could be there. Here you can count 1, 2, 3, 4, 7, means 6, 7 hairs will be present in the second one. And in the second subcontinent, two distinct hairs are there. So it is a black structure. These hairs are very stout. They are totally different from other bristles which are scattered all along the tarsus portion. But in case of malerkotliana, the sex comb pattern is different. In the first subtarsus segment, you can see a dark hair here. This is sex comb, followed by 3 hairs. So 1 followed by 3. And in the second subtarsus segment, you can see 1 hair followed by 2. So this is the pattern in case of malerkotliana, whereas a totally different pattern is there in case of bipectinata. So these are two features, that is the abdominal tip coloration and the sex comb pattern. These are two features which can be used to identify the two species. But their females, they cannot be. And suppose you collect flies from the nature, and in that collection you have bipectinata as well as malerkotliana females both present. So how can you know that this is female of bipectinata or malerkotliana? For that purpose you will have to put those females uh, individually in separate food vials, and you will have to wait till their progeny come. If these females have already mated in the nature, then they will be able to produce progeny in the food vials, and by observing their progeny, particularly their males, you can identify the type of female you have put in the vial. So that is the only way you can know that this female belongs to this particular species. Now here the two species means you can take bipectinata male flies and you can put them with the virgin flies of malerkotliana. So if these two flies are put together, means male of one species and female of another species, but this female has to be virgin. You have to collect this female from the stock of malerkotliana, then you can get the hybrids. So when you will observe these hybrids, what you will find that their male individuals hybrids have not been obtained in nature. This I told in the beginning, that people have collected flies from the nature, and since hybrids possess features of both the species, so such hybrids can be identified, but so far people have not been able to identify such hybrid males. But they can be crossed. These two species can be cultured in the laboratory and they can be crossed, and in such condition you can get the hybrid flies. So when you get the hybrid, in that case male individuals can be morphologically identified, and see in this diagram, this hybrid male will have black abdominal tip similar to the malerkotliana. But if you will observe its sex comb, its first tarsus subsegment will show the presence of sex comb pattern similar to Drosophila bipectinata. And in the second sub-segment, you will find uh, sex comb pattern similar to this malerkotliana, because I told you that in case of bipectinata, uh, two a group of sex comb will be there in the first segment followed by a single one in the second. But here in the hybrid you will have in the first subsegment of tarsus to sex comb, similar to bipectinata. Only thing is number of hairs will be little bit less, but in second subsegment the sex comb pattern will be similar to the malerkotliana sex comb pattern, that is 1 followed by 2. So this way the hybrid males can be identified, but they are females, and these males are sterile. See, the hybrid males, they are always sterile. They are not reproductively fit. If you dissect them, you will find that their males are sterile, but their females are fertile. This you can prove by crossing them with either of the parents. Means the F1 females or the hybrid females can be back crossed. They could be crossed either with bipectinata male or with malerkotliana male to get the progeny. So this is another feature you should understand, that F1 females, they are fertile. Now, for doing further experiments, we can actually have four options. Four different kinds of crosses can be made. This is one specific planning which is quite important to understand, and please be careful while these four different kinds of crosses are going to be made. You please get acquainted with them. There is one experimental plan in which we call as male choice experiment. See, male choice experiment means it is the choice of male to mate with different kinds of female. That is why it is called as male choice experiment. So what we do here, that we take male of one species and female of its own species as well as a different species. So here this male of species A will be crossed with, this will be crossed with females of species A, means its own type, as well as females of a different species, that is species B. So here in this particular case, what you can take suppose male of Drosophila bipectinata, and then in the mating chamber, these males will be introduced with uh, females of Drosophila bipectinata as well as females of Drosophila malerkotliana, means that is a different species. So in a single mating chamber, we use mating chamber which is a small wooden chamber. On the upper side of it, a transparent glass is there, so you can observe the mating pairs, and there will be one area through which the males and females can be introduced. So all these four experiments could be executed in the mating chamber, and as I told, the first one is male choice experiment, in which male of one species could be put with the female of its own species as well as a different species. The other experiment could be female choice experiment, where it will be the choice of female either to mate with male of its own species or male of other species. So here the female of species A will be crossed with male of species A plus male of species B. Means in a mating chamber you can have this setup. Suppose you have females of Drosophila bipectinata, and then you have also female of male of Drosophila bipectinata and male of Drosophila malerkotliana. Now this is up to this female that whether she is allowing its own male to mate with her, or she is permitting male of a different species to mate with her. That is why this is called as female choice experiment. In the third situation, it is no choice experiment, in which male of one species will be put with the female of other species. So there is no other choice, means this male will have to either mate with this female or will have to discriminate it. And exactly the same thing occurs with this female also. She has means no option either to allow this male or not to allow. So this is a no choice experiment. And then the fourth experimental plan could be multiple choice experiment, in which you will have male of species A as well as species B, and they will be put with the females of species A as well as species B. So here this male of one species has, suppose species A has choice either to mate with the females of its own species or females of other species, and likewise species uh, B male, they have choice to mate either with the means alien female or with its own female. So means these four different kinds of experiments could be set up, but we prefer to set up the first one. Most often we use this male choice experiment, in which we take male of one species, like we take male of Drosophila bipectinata, and then we keep females of Drosophila bipectinata as well as malerkotliana, and see that what amount of mating occurs among them. But one thing here itself I want to make clear, that if there is mating between male of A and female of A, then this will be referred as homogamic mating. Homogamic mating means mating between males and females of the same species. It will not be called as homogamitic, because homogamitic and heterogamitic are different terms. They are related with gamete formation. When we say homogamitic, sex, it means a type of sex which is capable to produce all the eggs of similar types. Heterogamitic sex means a particular sex which is capable to produce, to generate two different kinds of spermatozoa, or two different kinds of gametes. So we use such term while studying sex-linked inheritance pattern. Heterogamitic and homogamic terms are used over there. But here you will be using the term homogamic matings, that is mating occurring between similar type of members. And if mating occurs between two different kinds of members, then that will be referred as heterogamic matings. So if mating occurs, once again let me tell you, between a male and a female, it will be homogamic mating, and if it occurs between a male and b female, then it will be referred as heterogamic matings. Now come to the experiment. What experiment we do? Actually we put in the food vials or food bottles the Drosophila bipectinata male flies along with its own females as well as females of malerkotliana, and we are able to uh, then analyze the percentage homogamic and heterogamic matings. So here is an experiment. Male choice experiment was performed by keeping equal number of females of Drosophila bipectinata and Drosophila malerkotliana with males of only Drosophila bipectinata. So definitely it is a male choice experiment, because here male of one species is being put with females of two different species. Based on the observations of this experiment, percent homogamic and heterogamic matings was calculated, and the result is given below. Find out isolation index. So this is one parameter which we can calculate, that is isolation index, represented as II. Percent homogamic matings, that is given here in this data, that is 90%, means 90% A males mate with 90% uh, its own species females, that will be homogamic mating, and it is only 10% mating which occurs between the Drosophila bipectinata male and Drosophila malerkotliana female. Now there is a question. You people might be thinking that how this homo and heterogamic matings could be known. This is a simple thing to observe. Listen me very carefully. What will happen if suppose you have taken 200 females and you have put them with 100 males of Drosophila bipectinata. So 100 males of Drosophila bipectinata are there, and they are being put with the females which are 200, means 100 bipectinata females and 100 malerkotliana females. You will allow them to remain in the food bottle, suppose for 5 days, because you hope that in these 5 days matings might have occurred. After that, what you will do, you will discard all the males, means all those males will be thrown out. Only these 200 females will be taken, and they will be put in individual food vials, because you don't know those females that to which species they belong. They are morphologically similar, so you will be putting them in separate food vials, and then you will observe their progeny. If a Drosophila bipectinata female has mated with its own male, because that is the male present over there, then bipectinata flies will be available in that food vial. And if you find that in the wild hybrids are there, because Drosophila bipectinata, when it will mate with malerkotliana female, then you will have hybrid, and hybrid you can identify by the presence of a black abdominal tip and the sex comb pattern. So accordingly you can know that it is a heterogamic matings, and definitely we observe less number of heterogamic mating than the homogamic matings. Drosophila bipectinata male will prefer to mate with its own type of females. So naturally homogamic matings will be comparatively very high than these heterogamic matings. So if this kind of experiment is performed, and if you have this result, that is 90% homogamic mating and 10% heterogamic mating, then how you will analyze this result? What is there? This isolation index can be calculated by using this formula, that is percent homogamic matings minus percent heterogamic matings divided by percent homogamic matings plus percent heterogamic matings. So you know that 90% homogamic matings is there. So 90 minus 10. Here it will be 90 plus 10. Only those values will have to be put here. And if you find, and this value isolation index, it ranges from 0 to 1. If you find this value 0, it means this indicates no sexual isolation. It means homo and heterogamic matings are same. If homo and heterogamic mating, suppose 50-50% is there, so 50 minus 50, it will be 0, divided by 50 plus 50, 100, so final result will be 0. So if 0 is the isolation index value, then that indicates that there is no sexual isolation, means both species have not sexually isolated at all. And if you find this value to be 1, then that indicates that the two species are completely or sexually isolated, means they do not show reproduction in them, and there is complete reproductive isolation, particularly the sexual isolation is there. Ethological isolation is there. They do not like to mate with each other at all. So only homogamic matings will be there. In such case, this value will be coming 1. And if this value, isolation index value, is very close to 1, that is 0.85 or zero point 0.8 or zero point 0.9 or above zero point 0.9, then you can say that there is a strong sexual isolation. So on this very basis, you can infer that what kind of a result is there. And in this particular case, uh, I have made calculation, as I told, 90% homogamic matings are there. So 90 minus 10, and 10% heterogamic matings have been recorded in the question, and that will be divided by 90 plus 10, so it comes 0 0.8. So this indicates that there is a strong sexual isolation between these two species, like Drosophila bipectinata and malerkotliana. These two are although sympatric and phylogenetically very close species, and that is why sometimes mating may occur if they are being reared in the laboratory conditions together. And this value 0.8 means 80% sexual isolation is there. So isolation index value, it is really high, indicating that there is a strong sexual isolation. So Drosophila is a particular kind of genus which comprises hundreds and hundreds of species, some of them very closely related. They have common phylogenetic descendants, and still today some kind of uh, means mating may occur between some of the species. So in such cases, this kind of experiment can be planned, and isolation index values can be observed, can be calculated, and further analysis could be performed.